Welcome back everyone. So now we're going to talk about tetracycline-induced expression, TET-on and TET-off expression systems. So, what are they? Well, when we want to express a gene or a protein in a mammalian cell, a eukaryotic cell, there are two ways we can kind of go about that. That is overexpression, where the protein will just be produced all the time, or a more controlled approach, which is dependent on the presence of another molecule or signaling operator. So, TET-on and TET-off, tetracycline on and off signaling systems. As the names suggest, allow us to signal either in the presence of tetracycline or the absence of tetracycline, both of which achieve the same thing. It just allows us to have either the presence or the absence as the positive cause of transcription further down the genome. So what does that look like? Well, in TET, on. Oh, and I should also mention that tetracycline has derivatives which have the same effect. So in this case, with the Memphis meets pattern, we're discussing doxycycline, which is a derivative of tetracycline. It's similarly able to bind to RTA and TTA, which is the TET-on activator and the TET-off activator. So, we look at TET-on. So in the absence of tetracycline and doxycycline, we have our TTA activator. It's got no doxycycline in it, and it does not transcribe, does not permit the transcription of this part of the genome. In a moment I'll discuss how this part of the genome is created. Our alternative situation is we have our TTA, we have our doxycycline, and that permits the transcription of our genes further down the line. Now, this has been invented some time ago, and when we talk about expression systems in mammalian cells, eukaryotic cells, this is really the first system that comes up. This is the first well-controlled system that allows us to make sure that we're expressing genes in mammalian cells in a controlled and effective way. I'm going to go to red now. I'm going to go to red. So what's the alternative? TET-off. TET-off. And so, we're going to start on the left where we're not transcribing. And in that situation, we do have tetracycline, right? But in TET-off, does not cause transcription, because this binding operator does not permit transcription. Binding operator with TET does not permit transcription. So then of course we have the inverse, where we do not have doxycycline, and it does permit transcription. Pretty straightforward. So, the other transcription control vector that Memphis Meets is using the estrogen mutated approach. And when we look at ways to control specific genes in mammalian cells, these are kind of the two things that come up. There are different ways to control gene expression. There are different conditional on and off switches. But these are the two that really give you they're the ones that first come up. These are the first ones that you find out about when you're looking at expression. So it's not that special. It's not that special that Memphis Meets are using these two controllers to control their behavior of the mammalian cells. I'm going to pause this and then I'm going to come back and then I'm going to explain estrogen. All right, so estrogen receptor. estrogen receptor. So very much like the other inducible expression system that we discussed, TET on, TET off, the estrogen receptor system is inducible in the presence of a small molecule. So how does this whole thing work? Well, you'll remember similar diagrams for this. So we have MyoD binding site, with our target gene, and we have our myoder, and we have our small molecule, pictured here as a large molecule, as 17-beta-estradiol. 17-beta-estradiol. So, 17-beta-estradiol is a small molecule. Only in the presence of 17-beta-estradiol, do we get the expression of our target gene for this particular pathway. Now, the target gene, in this case, is MyoD. In the case of the O2H cell line, MyoD naturally causes differentiation. So what that means is, when we express this MyoD, causes a signal cascade. I might go through the whole thing, which eventually causes differentiation. So let's look at it from a genome perspective. So, yep, you have your genome. This red line's genome, right? Within the genome, there are coding sections. And there are binding sections. So, what we're doing is ectopically expressing the MyoD gene and associated proteins. So what does that mean? So normally, when we have our non-differentiated stem cell or myoblast, it's going to be expressing genes that are cohesive with its current state, that makes sense for that stem cell. It's expressing genes correctly. Now, like I said before, ectopic expression means that it's going to be expressing genes that are not correct for its current state, right? So what that means is, if we express MyoD, right, the MyoD is going to bind to a binding site. MyoD binds to a binding site, and then it causes, I'm going to need a new pen, it's going to cause the expression of a whole new set of proteins. All new proteins. New proteins, right? So the expression of these new proteins, right? Now what do those new proteins do? Those new proteins bind to our genome in new places. They bind to our genome in new places, causing a cascade of expression. So these new proteins, which have been expressed by the binding of 17-beta-estradiol to the myoder protein, which is in solution, onto the myoder receptor, causing the expression of the target gene MyoD, causing the expression of the MyoD protein, causing the binding onto our genome at a specific place, which is important for the differentiation of cells. These are places that would be expressed in differentiated cells. So MyoD binds to sites that are specific to differentiated cells, causing the expression of proteins which are important for differentiated cells. So that's the cascade that causes MyoD to differentiate cells. The only thing that we're doing that's different to the normal process is we're hijacking it by the expression of our 17-beta estradiol, otherwise known as E2. I'm going to write that. E2. E2. And that causes the direct ectopic expression of MyoD, which causes a cascade of effects making our cell transition. And that is how we ectopically express MyoD.